Welcome to Bioratna. Those of you who are watching me for the first time, let me introduce myself. I am Chandana Sengupta, having 18 plus years of teaching experience. I am honored to be recognized as Excellent Educationist by HRD Ministry, Government of India. Few of my suggestions have been incorporated in Standard 11th and 12th NCERT Biology. Many of my students have taken admission in government medical colleges, and every session for 12th board, I have given my bio toppers with 98-99 marks. But my real introduction is that I love my subject, of which I am a continuous learner, and I love and care for my students. Today's topic of discussion is monohybrid cross. So, we are starting with monohybrid cross, which can be defined as, let us first write it in a slightly wrong way so that it can register properly what is the right way to write it. Monohybrid cross, it is the inheritance of, it can be defined as the inheritance of single character. Inheritance of single character. But is it possible that only a single character is transferred, inherited by the offspring? The offspring inherits all the characters. It is simply the study of, inheritance of a single character. This is one thing by which Mendel, the father of genetics, he differed from his successors or the contemporary geneticists. What they were doing? At a time, they were studying all the characters which were inherited by the offspring and matching it with the characters of the parents. Here, what Mendel did, he studied, at a time he was studying just one inheritance of one character called monohybrid cross. Then for other characters he shut his eyes closed. अभी नहीं देखना है, पहले एक character का देख लेते हैं. Then he started with two characters, study of inheritance of two characters, that is dihybrid cross. And similarly, trihybrid cross was also performed by him. So we can say that monohybrid cross is the study of inheritance of a single character. Now, about Mendel. Mendel, we call him as father of genetics, as he is called father of genetics. So naturally, students think that he was the first one to perform the experiments on genetics. It is not so. There were many geneticists before him, at the same time with him, that they were performing the experiments. But Mendel, he is called father of genetics because for the first time he used mathematical logic and statistical analysis also for this biological hybridization experiments. And then he came to some conclusions which can be put into some concrete principles कि हाँ ऐसा अगर होगा तो ऐसा ही होता है. And those principles, even today, they stand true to the test of time. Of course, there are variations, of course, there are deviations from the rule, but they do not contradict what he has said. You have to remember certain dates. Mendel was born in 1822 in Austria. ऑस्ट्रिया में इनका जन्म हुआ था in a very poor peasant family. In 1856, he started his experiments on pea plant, garden pea, Pisum sativum. The plant is garden pea, Pisum sativum. In 1856, he started the experiment and did it for a span of 7 years. 7 साल के लिए किया. So, it is 1863. After 3 years, his work was published. So, it is 1866. Then Mendel dies in 1884 without seeing that his work has been recognized. 16 years after his death, in the year 1900, it was rediscovered. And then it was republished, and rest is history. And since then we call him the father of genetics. The days you have to specifically remember is this one, his birth date, that is 1822. These 7 years, you saw at first I fumbled while writing 1856, and then I could write it very fast. How? It is 1856. I always tell you that you have to follow some tricks to remember all the things. आप 1856 को कैसे याद रखोगे? आप पहले शुरू करते हो 5, 6, add 1, that is 7, and it is 8. After that, it is a child's play. इसमें आप 7 साल जोड़ देते हो तो 1863. This year is very important. The year of publication of his experiment, and then he dies in 1884. That is not, of course, significant for your study purpose, but it was rediscovered in 1900. 1900 is easy to remember. So this is about Mendel. He selected the plant which is Pisum sativum, and on it he carried out the monohybrid cross. Monohybrid cross, that to watch the inheritance of a single character. This Pisum sativum, it has many characters which are in contrast to each other. Hybridization में आप क्या करते हो? एक parent tall है, एक parent dwarf है. तब आपने क्रॉस किया, तब तो आप देखोगे कि ऑफस्प्रिंग टॉल है कि डॉर्फ है. If all the pea plants are tall or all the pea plants are dwarf, then what is the use of doing the hybridization experiment? But in this Pisum sativum, contrasting characters were available, out of which Mendel selected 7. We are going to see what are these 7 characters for which Mendel performed the monohybrid cross. Before that, few more characters why he selected Pisum sativum. Because this plant, it is normally pure. आपको एक pure material पहले चाहिए to start with. Why it is pure plant? Because it does self fertilization. It is a bisexual flower जिसमें stamen और pistil corolla के अंदर enclosed होते हैं बच्चों. So there is no chance of cross fertilization. It is pure form. Second, normally it is doing self fertilization. So when we try to do hybridization experiment on it, it might reject it, but it does not. Well and good. ये रिजेक्ट भी नहीं करता. फिर इसके पास कॉन्ट्रास्टिंग कैरेक्टर्स भी है जिसके साथ हम टेस्ट करके इनहेरिटेंस ऑफस्प्रिंग में देख सकते हैं. देन इट हैज़ अ वेरी शॉर्ट लाइफ साइकिल ऑल्सो. इट हैज़ अ शॉर्ट लाइफ साइकिल ऑफ फ्यू मंथ्स. तो आप 1 जनरेशन, 2nd जनरेशन, 3rd जनरेशन जल्दी जल्दी ग्रो कर सकते हो टू सी द इनहेरिटेंस ऑफ अ पैटर्न. Then it can be very easily grown. So it has so many characters for which Mendel selected the garden pea Pisum sativum for his experiment. Let us see the 7 contrasting characters. 7 contrasting characters found in Pisum sativum. So 7 they are: 1, 2, 3, 4, 5, 6 and 7. Okay, which character? And the two contrasting traits. Fine. Trait का नाम सीधे नहीं लिख के what we do, we write one character as dominant, another character as recessive. What is this dominant and recessive? We are going to see. Let us start. The first one. It is the height of the plant. Height. Dominant is tall. Recessive is नाटा वाला, dwarf. बता ही दे आपको कि dominant और recessive किसे कहते हैं? When you cross a pure tall parent plant which you have selected with a dwarf pure plant, then in the first generation 100% offsprings show only one character. दूसरा दिखता ही नहीं है, तो जो दिखता है उसे dominant कहते हैं. So if we take one tall plant, one dwarf plant and cross it, then 100% offsprings with the character which they show, we are going to write it in this column, dominant. Fine. Next is flower. Flower color. It has two types of flower, either violet, violet is the dominant one, or white. Fine. Flower का और एक character, flower position. Flower position, it is dominant is axial and recessive is terminal. What is axial and terminal? You remember na? The flower which is present as the terminal position, that is called terminal, and the one which is present at the axis, axial. So axial is dominant. Now come to the next character, that is seed color. Seed color, यहाँ बच्चे तुरंत गलत करते हैं ना, बहुत बार गलत करते हैं. They think that green seed is the dominant one. No, yellow seed is the dominant and green seed is the recessive. Now come to another character of seed, that is seed shape. Round shape is dominant, ऐसे round है shape उसका, and wrinkled shape, that means अगर वो ऐसे 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 this type of shape it is having, that wrinkled seed is recessive. और दो बचे, कौन सा? pod. pod किसे कहते हैं बच्चों? वो कवरिंग जिसे आप छीलते हो जिसके अंदर सीड्स होते हैं, that is the pod. The first is pod color. Dominant is green and recessive is yellow, just opposite of seed color. Another for pod shape, inflated is dominant. Inflated मतलब normally जो आप खाते हो ऐसे shape का जो पी होता है ना, that is inflated. And recessive is constricted. Constricted मतलब कैसा? ऐसे ऐसे seed का shape ले लेता है ना वो pod. Then it is called constricted. So these are the 7 characters for which Mendel performed his crosses, out of which we are going to see the monohybrid cross. जब वो height का देखेगा, when he will study the inheritance of height in offspring, at that time he will not observe the other characters. That simple, monohybrid cross. Understood? Now let us start with the experiment and what results he obtained. So now we see the experiment first as a layman, what experiment was done and what results were obtained. Then we will see the genius at work, Mendel, the statistical analyzer and mathematical logic भी जिसने लगाया था. So first the simple one. Remember one thing, this monohybrid cross is the base of all the genetics. जितना बाद में जितना आप करते जाओगे ना, जब भी आप नहीं कर पाओगे, कम बैक टू दिस मोनोहाइब्रिड क्रॉस. मोनोहाइब्रिड क्रॉस जिसने अच्छे से कर लिया, दिस इज द बेस. उसके ऊपर फिर उसकी कहानी आप खुद से अपने आप बना सकते हो, स्टेप बाय स्टेप. इट विल बिकम सो इजी फॉर यू. लेट अस सी. This is a tall plant. He took a tall plant and this sign is for cross. Tall plant crossed with a dwarf plant. Fine. These are the two parents. That's why we write P. This should all be in the same line ना. यहाँ ऊपर P लिख दिया, नीचे P लिख दिया. Don't do that. Then when these are crossed, then the first filial generation, it is called filial. This is first, F for filial. The first filial generation, he got 100% offsprings as tall. 100% offsprings tall. You know that pea plant, this Pisum sativum, it normally reproduces by सेल्फ फर्टिलाइजेशन. अब आपने जबरदस्ती का क्रॉस फर्टिलाइजेशन किया, सो नाउ यू विल थिंक कि क्या हुआ, ये डॉर्फ गायब हो गया क्या? दैट फैक्टर, दैट समथिंग विच कॉजेज द प्लांट टू बी डॉर्फ, हैज दैट कैरेक्टर वैनिश्ड, डिस्ट्रॉयड? व्हाट हैपेंड टू इट? ये जब आपको करना है तो आप फिर से क्रॉस तो नहीं करोगे. आपको उसी ऑफस्प्रिंग में, उसी इंडिविजुअल के अंदर आपको देखना है कि भैया आपके अंदर क्या है, क्या आपके अंदर अभी भी ये डॉर्फ का फैक्टर जिंदा है. अंडरस्टूड? दैट इज द रीजन व्हाई हियर इट इज नॉट क्रॉसिंग बट इट इज सेल्फ. सेल्फ मतलब फ्लावर तो है ही क्लोज्ड, नॉर्मली वो करता ही है सेल्फ पॉलिनेशन. पोलन फ्रॉम द सेम स्टेमेन विल फॉल ऑन द स्टिल ऑफ द सेम फ्लावर. इससे उसे in next generation, that is F2 generation, he obtained tall as well as dwarf in the ratio of 3:1. आ गया वापस. That means that dwarf, though it was recessive, दूसरे के प्रेजेंस में वो खुद को एक्सप्रेस नहीं कर पाया, but it kept itself alive. It was very much there and it expressed itself in the F2 generation in the 3:1 ratio. This is the famous ratio, the 3:1 ratio, what is called monohybrid ratio. इसी को कहते हैं monohybrid ratio. This is called 3:1 ratio. This way if you say that it will be wrong now. पूरा बोलना पड़ेगा. The 3:1 ratio which is obtained in the F2 generation of monohybrid cross. I repeat again. The 3:1 ratio, point number 1, obtained in F2 generation, point number 2, in the monohybrid cross, point number 3, is called monohybrid ratio. ऐसे तो देख लिया. अब इससे कोई क्या प्रिंसिपल बताएगा, क्या करेगा? But now our genius gets into his work. Mendel at work. The same experiment ना, फर्स्ट वो एकदम सिंपल सा लगने वाला, छोटे बच्चे का क्लास का लगने वाला, उसको अच्छे से समझ लीजिए. उसके बाद उसी को हम लोग अब मॉडिफाई करते जाते हैं. देखिए कैसे. द पेरेंटल जनरेशन, इट वाज, वन वाज प्योर टॉल एंड अदर वाज प्योर डॉर्फ. फाइन. नाउ Mendel thinks कि okay, there has to be some factor which is making this plant tall and another factor which is making this dwarf. Let us take some letter for it. X ले लेते हैं, प्रॉब्लम हो जाएगी ना. 7 कॉन्ट्रास्टिंग कैरेक्टर्स है, सबके लिए x x x x लेने से काम चलेगा. तो एक बड़ा सिंपल सा जीनियस सा तरीका उन्होंने अपनाया. What he did? Out of these 2 characters, the one which is dominant, which expresses itself in F1 generation, उसका first letter लेके ये हो गया उसका capital T, इसका factor. And here it is not D, but this is also T, but small t. Means automatically it shows that this one is recessive, which is having small letter, and this one is dominant, which is having capital letter. Now, it's sexual reproduction, product of fertilization of two gametes from two parents. So, here it is 2 capital T and here it is 2 small t. Simple. Now, there is one extra step to form the gametes. Here what now he is doing, here there are 2 capital T, the number is made half in the gamete, so this becomes only single capital T, and here it is single small t. Then only when these two will fuse to form the gamete and form the offsprings of the next generation, then in them also the same number will remain. तो ये हो गए गमीट बन गए दोनों के, दोनों ये तो प्योर था, दोनों कैपिटल टी थे, तो एक ही तरह का गमीट बन रहा है कैपिटल टी वाला. तो 100% are capital T, here 100% are only small t. So the first generation, the first filial generation, here these two are combining, so this is capital T and small t, and these all are 100% tall. Fine, इतना तो हमने वहां भी कर लिया था, वही है, बस अब देखिए इसका. Now this F1, it is not crossed. I have told you the reason. This is done selfing. When we are doing selfing, both the male gamete and female gamete are from the same flower, so they will be having this same genotype. So it is capital T small t and capital T small t for both. Let me draw this dotted line to separate the male and female. Now we will form gamete for this. यहाँ पे this is not pure. This is both are having one capital T, one small t. So 50% gametes will be having capital T's, 50% will have small t. Similar on this side. यहाँ पे एक मेल का निशान लगा दे, यहाँ पे एक फीमेल का निशान लगा दे. Now these gametes will fuse and form the F2 generation. कैसे बना? देखें. See, if this gamete fuses with the first of this one, then it is capital T, capital T. But if it fuses with this one, then it is capital T, small t. इसके दोनों possibilities खत्म हो गए. Now let us take the next one. This small t, it is joining with this capital T. So capital T is always written first, na? So this is capital T, small t. And this last one, it is small t, small t. Take care बच्चा, ऐसे देखने में आसान लगता है ना, but while drawing you have to take care. जिस पॉइंट पे आपने मिलाया, देखो ये चारों मिलने वाले पॉइंट्स भी एक सिंगल सीधे लाइन में है. At the same time, जिस पॉइंट पे हम लोग ये मिलते हुए दिखा रहे हैं, the 2 points, exactly at the same point you are writing the terms. यहाँ पे आपने मिलाया, यहाँ आपने लिखा. Students do that very frequently. Don't do that. This mess up the whole thing. आपके ब्रेन में भी ठीक से रजिस्टर ऐसे नहीं करेगा. एलिमेंट तो खत्म हो गया, लेकिन अब इसमें से बहुत इंपॉर्टेंट बहुत सारी चीजें निकल के आएगी. क्या? देखिए, here out of these 4, only this one, it is pure tall, both capital T, whereas this 2, they are impure tall, heterozygous tall, capital T as well as small t. And the last one, this is again pure, but it is having small t, small t. It is different from the pure tall. This is 1. So this is the ratio of 1:2:1. जो उसमें कौन से फैक्टर से देख के हमने जब ये ग्रुपिंग की है, then we have got the ratio 1:2:1. So this is called genotype. उसका जीन का क्या टाइप है, वो हमें बता रहा है. So 1:2:1 is the genotype. अगर हम उसको बाहर से देखते हैं तो वो हमें जीन थोड़ी दिखाई देने वाले हैं ना. ये all these three they will look tall and this one is dwarf. One is dwarf and three are tall. So this 3:1 ये उसका मॉर्फोलॉजिकल कैरेक्टर, बाहर से उसका फिनोटाइप दिखा आपको. सो दिस इज कॉल्ड फिनोटाइप. बहुत क्वेश्चंस आते हैं इसमें और इतने सुंदर घुमा के मजेदार क्वेश्चंस आते हैं ना, यू विल फॉल इन लव विद जेनेटिक्स with simply solving the problems based on it. You don't have to worry about it. It is actually अभी तक मुश्किल लगा कुछ? The whole genetics मतलब पहला चैप्टर लास्ट तक, सेकेंड चैप्टर, this is such in a flow ना. लेकिन the first monohybrid cross आपको एकदम फिंगर टिप पे होना चाहिए. It looks very simple और वो फिर वो बाकी सबको सिंपल बना देता है. और एक तरीका है, ये यहाँ तक आपने गमिट्स बनानी है, ये यहाँ तक आपने गमिट्स बनानी है, ये गमिट्स बनानी है. इसके बाद ये गमिट्स कैसे ज्वाइन होंगे, इसको इस तरह से नहीं करते हुए देयर इज अनदर मेथड दैट इज कॉल्ड पुनेट स्क्वायर. इस पुनेट स्क्वायर a method developed by Pune. We draw a square. हाँ, एक स्क्वायर बनाया. अच्छा, ये इसमें how many blocks we are going to draw, that depends upon how many male and female gametes are there. So one is this we have written vertically, one we have written horizontally. Gametes हो गए. जब gametes हो गए तो उसका फ्यूजन प्रोडक्ट लिखने में क्या देर है, कर दीजिए. This T and this T, बाहर वाले ये दो मिल गए. This is capital T and capital T. अब इस बॉक्स में आइए. This is small t, this is capital T. So we write capital T first and small t. This box, ऊपर कौन है? Capital T. साइड में कौन है? Small t. Capital T, small t. Last, ऊपर कौन है? Small t. साइड में कौन है? Small t. So this is the only one which is the pure dwarf, this one. The same thing you will get. Many times it is asked कि पुनेट स्क्वायर में ही बना के हमें दिखाइए, and many students they find this simpler. It becomes much simpler. ऐसे मोनोहाइब्रिड क्रॉस में तो दोनों सिंपल है, but when you move to dihybrid cross, वहाँ पे इस मेथड से करना पॉसिबल नहीं होता. There we have to follow the पुनेट स्क्वायर. तो पुनेट स्क्वायर को सिंपल वे में हमने मोनोहाइब्रिड क्रॉस में बना लिया. डन, खत्म तो हो गया. Now there are few terms which come out of this cross. कौन कौन से टर्म्स है? सारे के सारे बहुत इंपॉर्टेंट ना, कहीं पर भी पूछा जाएगा. आपको बोर्ड में पूछ लिया, आपको नीट में पूछ लिया, क्या क्या है देखो. Here you see, ये वाला इंडिविजुअल देखिए या ये वाला देखिए. It is having both capital T, it is having both small t. We are using the term pure because आप लोग अभी पढ़ रहे थे तो सिंपल वर्ड यूज हुआ था. But the word which we use is homozygous for the pure one, having both the same allele. So this is homozygous, this is homozygous. अब F1 में आ जाइए. देखिए ये F1 वाले offsprings है. They are having two different types of alleles. So they are called heterozygous. They are called heterozygous. ये हो गया. Monohybrid cross I have already told you. This genotype ratio 1:2:1 ratio obtained in F2 of monohybrid cross when we group them according to the alleles present, that is genotype ratio, and the 3:1 ratio is the phenotypic ratio or the monohybrid ratio obtained in F2 generation of the monohybrid cross. इतने हो गए. alleles अच्छे से याद रखना. The two forms, tall dwarf, two forms of the same gene, the same character are called alleles. Okay, so this was it. Monohybrid cross done. Hope all your concepts are clear. If you are new to my channel, then do subscribe and hit the bell icon so that you get all my notifications in time. Thank you for watching the video.